Hello everyone. Today I will be going through the Combined Science Trilogy Biology Paper 1, Higher, and it is the 2022 paper. So, question 1. Bacteria can cause a variety of diseases in humans. What are the two similarities between a bacterial cell and an animal cell? Tick 2 boxes. So a bacterial cell and an animal cell both have a cell membrane and both have a cytoplasm. Remember, bacterial cells do not have a nucleus. Bacterial cell has a cell wall, but an animal cell does not have a cell wall. Plasmids are only found in bacterial cells and not in animal cells. Salmonella food poisoning is caused by bacteria in food. Give one symptom of salmonella food poisoning. Do not refer to vomiting or diarrhea in your answer. So if you have salmonella food poisoning, what would be the symptom? What would you see? Make sure you're not mentioning what's already here. So if you think back to what's on the spec, it says fever. Okay, and it's only 1 mark, so you need to write one thing down. Now be careful, do not write down stomach pain because you won't get a mark. You can write down stomach cramps. Or you can even say abdominal cramps. For fever, if you don't have fever, you can say high temperature, you would still get a mark, same thing. Next question. What is the name of the first antibiotic developed? Penicillin. A child with a severe bacterial infection was given a course of antibiotics. Figure 1 shows how the concentration of live bacteria in the child's body changed when taking the course of antibiotics. So this is where the course was started, so at 0. Concentration of live bacteria is high. Despite the child taking antibiotics, it's still increasing, and then it gradually decreases, and that's your time in days. The concentration of live bacteria in the body continued to increase after starting the course of antibiotics. Suggest one reason why. And here, have a think. As soon as you start taking antibiotics, you won't see the effect immediately. It takes time, and that is your answer. So you say it takes time for the antibiotic to work. After 3 days of taking the antibiotic, the child felt better. There was still bacteria in the child's body. Why did the child feel better? So again, look at the graph. Bacteria had become immune to the antibiotic. That's wrong. The child has become resistant to the bacteria. There were fewer toxins in the body than at day 0. Remember, bacteria produce toxins. And if you're taking antibiotics, antibiotics will kill bacteria. So over time, your toxin levels should decrease, which is what you can see on the graph, and eventually it will be 0. Suggest why doctors do not give antibiotics to patients with minor infections. And that is to reduce antibiotic resistance. Figure 2 shows blood viewed using a microscope. Okay, so let's just quickly go through each of these components. So let's start off with these fragments that you see here. These are broken up cells. So D must be platelets. B must be white blood cells. White blood cells are larger than red blood cells and they have a nucleus, an irregular sized nucleus, and you can see that here. A must be your red blood cells, and then C is your straw coloured liquid, which is your plasma. A vaccine will stimulate the production of antibodies. Which part of the blood in figure 2 produces antibodies? Well, we know it's the white blood cells. So let's go back to the diagram, and it is B. Which part of the blood starts the clotting process? Well, you know that's platelets. So you would tick D. Question 2. This question is about cell division. Write the biological structures from the box in the correct order of size. Start with the smallest first. So obviously cell is larger, then the cell has a nucleus, and within the nucleus you have chromosomes, and a small section of your DNA is a gene. So the smallest would be your gene, largest is your cell, then chromosomes would go there, and nucleus would go there. So make sure all of that is in the correct order to get the 1 mark. 2.4. In humans, a fertilized egg cell contains 23 pairs of chromosomes. How many chromosomes will there be? Oh, sorry, I think apologies. That was the question. Okay, so 2.2. Name process A. So let's have a look. We've got a fertilized egg cell. It divides, obviously fertilized egg cell divides by mitosis. You end up with a ball of cells, which is an embryo, and then you get some of these cells changing into red blood cells, some change into muscle cells, some change into nerve cells. So these cells are specialized cells. This process of specialization is called differentiation. How many cell divisions are needed to form a 16-cell embryo from the original fertilized egg cell? So, you have a cell, it divides into 2, yeah, then each cell will divide again, so you end up with 2 going into 4. Each cell divides into 2, so 2 times 4, you would end up with 8 cells. 2, 6, 7, 8. If each cell divides into 2, so that's 8 times 2, you should end up with 16 cells here. So I've got 2 16-cell embryo, but the question says how many divisions are there? So let's count. 1 division, 2nd division, 3rd division, 4th division. So there's 4 divisions taking place. Okay, 2.1, 2.2. Okay, in humans, a fertilized egg cell contains 23 pairs of chromosomes. How many chromosomes will there be in each of the embryo cells? And the embryo cells are body cells. So they must have 46 chromosomes, or you can say 23 pairs. That should still get you a mark. Figure 4 represents a cell cycle for a human embryonic cell. Describe one change in the cell that occurs during each of the stages of the cell cycle. Okay, so here you're just stating down what is happening in each of the 3 stages. They've already named stage 2, which is mitosis. Stage 1, you can see this is the stage where our cells spend most of the time. So in stage 1, we say, there's quite a few things you can say. It's 3 marks, okay? But what I'm going to do is I'm going to go through more than one thing that you can write, just in case a 6 marker comes up on this, okay? So stage 1, you can say the DNA replicates. So if I have 46 chromosomes, those 46 chromosomes will replicate and I will end up with 92. Now think about this. If this is happening, then the cell must be increasing in size. Also, the organelles also increase in the number. So ribosomes will increase in number. Mitochondria will increase in number. So you can write that down. So you can say increase in the number of organelles. Or you can give an example of an organelle and say ribosomes, or you can say mitochondria. For DNA replicates, you can also say chromosomes replicate. Okay, and there's cell growth. Okay, stage 2, well, it's already named mitosis. So what exactly happens in mitosis? So you can say, you don't need to know the stages of mitosis, so prophase, metaphase, anaphase, telophase. You do not need to know that for AQA. That is Edexcel only. So for stage 2, you can say that chromosomes line up across the center of the cell. Okay, so if you think back, some of the nucleus disappears, your chromosomes line up in the middle of the cell. That will get you 1 mark. You can also say chromosomes are pulled to each end of the cell. So all chromosomes are pulled to each end of the cell. You can even say 2 nuclei form. So for this question, you only need to write one thing. But I'm just going to give you additional bullet points that you can write just in case this question turns up and, you know, as a 6 marker. Stage 3 is where your cytoplasm or the cell membrane divides to form 2 genetically identical daughter cells, which is cytokinesis. So for stage 3, if you just say cytokinesis, you should get a mark. Okay, or the cytoplasm divides. So if you have all of that, that should get you 3 marks. Okay, so 2.6, a 6 marker question. Cell division is important in the growth of multicellular organisms. Figure 5 shows the mean height of boys and of girls from birth to age 18 years. So take your time, have a look at that graph. Okay, so my bold line is girls, my dashed line is boys. Age in years, I'm going from 0 to 18 years. And then I've got my mean height in centimeters. Compare the growth of boys with the growth of girls. Use data from figure 5 in your answer. Okay, so if you think about it, this is a straightforward question. You don't need to use your own knowledge for this. The graphs here, use the graph and compare. A compare question means you need to write about the similarities and differences. So it's a comparative question. So roughly, more or less, 6 bullet points should hopefully get you the 6 marks. So general comparisons, and then we'll have a look at the rate. So we can say, so if you look at the graph, so let's have a look at, so for example, you can see that lines meet here, which is halfway here, so that's 11, age 11. So I can say girls and boys are the same height at age 11. And if you have a look at 11 to age 14, the girls' line is above the boys' line. So we can say girls are taller than boys, but you must write the age down, which is more or less between age 11 and 14. You can see boys, so you can see here that the growth of girls comes to an end, whereas the boys are still growing. So I can say boys carry on growing for a longer time than girls. And then you can go back and say, well, the girls stop growing age 14 and boys stop growing at, you can say, age 17 or 18. Age 13 or 14, and age 17 to 18. If we start looking at the growth rate comparison, so we can see that the rate of growth, okay, so we're looking at the gradient of a line almost. You can see that's more or less the same, the steepness of the line. So you can say the rate of growth is similar up to age 10 or 11, more or less. Say up to age 10. So how many bullet points do I have? 1, 2, 3, 4, 5. So if I put another bullet point down, then I've got enough. So I can say, what else can we have a look at? Girls grow, girls, yeah. So look at the girls line here. That's steeper than the boys line. So here we can say that there's an increased rate of growth in girls aged 10 to 13, because we're looking at that bit there in comparison to the boys. So increased rate of growth in girls aged 10 to 13 and in boys, because it's a comparison question, you must also look at the boys as well. So and in boys aged 13 to about 17ish. So increased rate of growth in girls aged 10 to 13, and in boys, that's the bit we're looking at. So if age 13, you can say age 14 to 17, that's where you've got increased growth in boys. So that should be sufficient. Okay, and remember, you don't have to write in paragraphs. It's actually easy to write in bullet points. I'm an examiner, so I mark, and it's very clear that the markers are just looking at your science. So feel free to write in bullet points. Students who write in bullet points are the ones who actually attain more marks and get a better grade. Writing in bullet points allows you to get your thoughts down and your answers down in a logical way. Whereas in paragraphs, you will start waffling. So 2.7, give one way that cell division by mitosis is important in fully grown animals. So be careful, it's in bold. It means the animals have fully grown, but why is mitosis still important here? How is mitosis being used here? So here, growth has occurred, so this must be about repairing. So you have to write down repair of tissues, or repair of organs, or replacement of cells, but not repair of cells. Okay, so repair of tissues, or you can say repair of organs. So question 3, this is digestive system, so it's B2. And something I've also mentioned in my previous videos, whenever you read a question, have a think. So especially if you're struggling and you can't remember what you revised, have a think. What is the underlying topic in this question? So amylase is an enzyme that digests starch. It breaks down starch. Which organs in the human digestive system produce amylase, the enzyme? Okay, so let's use a process of elimination here. Liver doesn't produce digestive enzymes, so we know it's not that one. So it can't be that one either. Okay, and then we also know that large intestine doesn't produce digestive enzymes. So we know it has to be the third one. And you all should know that amylase is produced by the salivary glands in the mouth, pancreas and small intestine. A student investigated the effect of pH on the activity of amylase. This is the method. Prepare amylase solution at pH 5. Mix the amylase solution with starch in a boiling tube. Remove a drop of the amylase starch mixture every 30 seconds and test it for the presence of starch. Record the time when all the starch has been digested. Repeat steps 1 to 4 using amylase solution prepared at pH 6, 7 and then 8. This is your required practical, enzyme and pH. What was the independent variable? I change. That's how you remember this. Independent, what am I changing in this experiment? Go to step 5, prepare solution. So I've got pH 5, I've got pH 6, 7, 8. So it must be the pH that I'm changing. Next question. Describe how the student would know when all the starch has been digested. So here, think. How do we test for starch? Well, we use iodine solution. And you should all know that when you add iodine solution to starch, it goes blue-black. That's only if starch is present. Now, starch is a long polymer. If I break these links, and obviously amylase would do that, you produce glucose. Okay? If you add iodine solution, now, as I say, all this starch has been broken down. You add iodine solution, it will not turn blue-black because there is no starch, because the starch has been digested. So, going back to the question again, describe how the student would know when all the starch had been digested. Iodine would not turn black. Or you can say iodine solution would stay orange, because that is the colour of iodine solution. Then you should be familiar with this graph. Figure 6 shows the students' results. So I'm sure all of you have done this required practically. You've plotted a graph and it looks like this. Okay, what was the optimum pH, the best pH for amylase? So for this graph, if you look at the y-axis, it's time taken to digest all the starch in minutes. The lower the time here, that is your optimum pH. So obviously it'll be the lowest point of the curve. That is 6.8. A scientist did a different investigation. This is the method used. Prepare amylase solution at the optimum pH. Mix the amylase solution with starch in a boiling tube. Measure the concentration of sugar every 10 seconds for 2 minutes. Figure 7 shows the scientist's results. Concentration of sugar, and then you've got time in seconds on the x-axis. How much time did it take for the amylase to digest all the starch? So you're looking at where the concentration of sugar starts to stay the same. So if you look carefully, that bit there, the line is slightly going up. That's where the flat line starts. So I would say 82 seconds. If you say, some of you might be, I know it's not, I mean I can't zoom in, but say if you said 80, you would still get a mark. There is a range for this, and it's 80 to 84. So if you're looking at this thinking, oh, I think it's 80, that's fine, you should still pick up a mark. Then it goes, determine the rate of sugar production per minute at 40 seconds. So here, rate suggests gradient of a line. So if you go back to this graph, it's the gradient of a line, and we're looking at the rate at 40, so it's this bit there. Now, I can't work out the gradient of this curved line. So what you do is you draw a tangent at that point. Okay, that will get you 1 mark. Okay, and then what you need to do is to draw a right angle triangle, roughly symmetrically sitting above 40. And just be careful where you draw your right angle triangle. Try to include full squares, because then it's easier to work out the change in y and then the change in x. So I'm going to go for if I go there, okay, and let's read the top. So there's my line. Let's read the top value there. So that's 3.5, that's 3.6, that's 3.7, and it's actually halfway, so I'm going to say that's 3.65, that's 3. So the change in y, okay, is 0.65, okay, and that would be 44. That would be 28. 44 take away 28 is 16. So the base of that triangle is 16. Gradient of a line is change in y over change in x. So 0.65 over 16. So 0.04. I'm just going to write the whole number down. 0.65 divided by 16. Yep, so that's my rate. Okay, so that's units per second. But the question, if you look at what it says here, arbitrary units per minute. So now I need to take this answer there, multiply that by 60. That gives me 2.4375. It doesn't say how many sig fig, it doesn't tell me how many decimal places, and I'm going to write the full number down. Best to write the full number down. If in doubt, put it to 2 decimal places, unless the question specifically says 2 significant figures or 3 significant figures. Okay? 3.7, explain how the structure of enzyme molecules is related to the effect of pH on the activity of amylase. It's quite a mouthful. This question is basically saying, how does the pH affect the structure of the enzyme? It's an explain question, so this should be underlying science. So the underlying science here is looking at the structure of the enzyme, so you must be writing about the lock and key theory. Okay, so again, we're going to go for 6 bullet points. Okay, so I'm going to start off by saying enzymes are protein molecules they have an active site. This active site has a specific shape. So we can say at pH values above or below optimum. So obviously in the question earlier on we said 6.8. So if it's below 6.8 or above 6.8, the shape of the active site will change, and I'm sure we all know what that means. Enzyme is now denatured. That means the substrate, which is the starch, can no longer fit into the active site. Okay, and we've already said because the active site has been denatured, so amylase cannot digest starch. And then finish it off, so the rate of digestion would decrease or eventually stop. Okay, that is it. Okay, so you've explained the effect of pH, whether it's below optimum or above optimum. You have stated what happens to the active site and then how that relates to digestion. So question 4, photosynthesis. So this is the last part of the biology topics. Photosynthesis is an important chemical reaction in plants. Why is light needed for photosynthesis? And that is to transfer energy. Or you can say photosynthesis is an endothermic reaction. Right, what is the equation for photosynthesis? So for photosynthesis, think, okay, I'm going to make oxygen and I'm going to make glucose, C6H12O6. So these are the 2 products you need to be looking for after the arrow. Okay, so can't be that one because I've got water there. Can't be that one. Formula is wrong. And it can't be carbon dioxide. And then you would tick the third one. A student investigated the effect of different colours of light on the rate of photosynthesis at room temperature. Student used pondweed. A piece of pondweed was placed in red light, blue light, green light. Each colour of light was the same intensity, so you're controlling light intensity. Describe how the student should make accurate measurements to obtain valid results for the rate of photosynthesis. So this is your photosynthesis required practical. So it's talking about having data there that you can rely on in order to come up with a conclusion. So when you set up this experiment, you won't start collecting the volume of gas straight away. You need time for the pondweed to acclimatize, get used to the conditions, start producing oxygen, and then you start measuring that oxygen. Okay, so you can say leave the pondweed in the light for a few minutes. You can say 5 minutes before taking measurements. It says obtain valid results. So that means counting oxygen bubbles won't give you accurate readings. It's better to use a gas syringe. Okay, so that should get you a second mark. And you can also say measure the oxygen collected using a timer. Or a stopwatch. And then whenever you have a question like this where it talks about how would you obtain valid results, always write this down. Repeat the measurements and calculate a mean. Scientists investigated the effect of different wavelengths on the rate of photosynthesis. The wavelength of light determines the color of light. Figure 8 shows the student's results, which is a bar graph. Figure 19 shows the scientist's results. So you've got a bar graph, you've got a line graph. Why are the results of the two investigations presented differently? Okay. Remember, whatever you plot on the x-axis is your independent variable. And what you're measuring, dependent variable, goes on your y. So here you would say the independent variable in the student's investigation is categoric, or the other word is discrete. What that means is I've got fixed groups, red, blue, green, whereas the independent variable in the scientist's investigation is continuous, because I have a range of values from 400 all the way down to 700. Or you can even say the wavelength is a continuous variable because I've got lots of in-between values. Suggest the range in wavelength of green light. use figure 8 and use figure 9. Okay, so green light. So I'm going to go to figure 8, look at green light. Okay, and read that value off. So that's 0.6. So the rate is 0.6. Okay, and I'm now going to find 0.6 on my figure 9. And it looks like it's right at the bottom here. So that's 2, that's 1, right here. And if you have a look, what wavelength do I have at the bottom? Close enough is this bit here. That's the lowest part of the graph that I can see. So if I was to read the values from here, I'm just going to make my life easy. I'm just going to go for 500. I'm going to go for 600. Okay. On the mark scheme, there should be a range. So if I have a quick look, so your range for the first value could be anywhere from 475 to 525. And for your second value here, that could be anything from 575 to 650. So the range is quite big. I went for 500, 600 because, you know, I want quick numbers. I don't want to waste any time trying to work out, you know, what half a square is and so on. I know it's the lowest part of this graph here. So I went for those two values because those values already exist. Okay, 1 mark. Right, this question is about tumours. Describe the similarities and differences between benign tumours and malignant tumours. It's 4 marks. Okay, I'm going to go through as many points as I can, just in case this question does appear in your summer exam, which is in a few days' time, on Tuesday. You've got more answers that you can actually put down. So let's start off with similarities. So put down a heading. Let's have a look at what both of these tumours have in common, and we should all know that these tumours are there because of uncontrolled cell division. And why do we have uncontrolled cell division? You can say it's results from mutations or changes in DNA. Both will have lump of cells, so can form lump of cells. Both are made up of abnormal cells. So these are your similarities. Differences. So the differences I'm going to write is in reference to malignant tumours. Or you can do the converse, the opposite, and write about benign. So every bullet point that I'm writing is in regards to malignant tumours. So malignant tumours are made of cancer cells, and benign are not. So this is a comparison question, so make sure you are then going back and referring to benign. Malignant tumours grow faster than benign. They invade neighbouring tissues. But benign don't. Benign tumours can spread. but benign stay in one place. Or here you can say that malignant tumour cells can travel in the blood, but benign tumours do not. And then there's the final bullet point leads on from this. You can say malignant tumours can form secondary tumours, but benign tumours do not. Okay, so just one more time, remember it's a comparative question. So you write about one tumour and then you must refer to the second one, because obviously you need to make the comparisons. You don't need to have all of that for 4 marks, okay? So I've just gone through as much as I can. So 1, 2, 3, 4, 5, 6, 7. I've given you 9 bullet points. In fact, you only need 2 similarities and 2 differences. The reason for going through all possible points is if this was a 6 marker, you would need additional things to write, and you've got that in front of you right now. Figure 10 shows data about skin cancer in males and females, 4 different age groups in the UK. It shows the number of new cases of skin cancer in 1 year, the number of people with skin cancer per 100,000 population in 1 year. So this is that side of the graph, and that is that side of the graph. New male cases, new female cases, and then make sure you look at the key here. Your dashed line is number of males, bold line is number of females. So 5.2. There are no new cases of skin cancer diagnosed in people younger than 15 years of age. Explain why. 2 marks, so underlying science. So under the age of 15, people haven't been exposed, or very little exposure to UV light or ionizing radiation. So little exposure to UV light. So explain question. So why? So less damage, so less DNA damage. Remember, DNA damage is what leads to cancers. Give 2 conclusions about the number of new cases of skin cancer. So you're looking at this side and you're looking at the bar graph. Okay, so for 2 marks. So if you have a quick look, you can see there are more females than males diagnosed each year up to aged 59. And then you can say that there are more new cases in males than in more new cases in males than in females from 60. You can also say there's a greatest number of people, or males or females, diagnosed age group 65 to 69. You can also say number of new cases increases in males up to the age 65 to 69. After that, you can see that it declines. Okay, the data for the number of people with skin cancer is given per 100,000 population. And again, I've seen this question in different papers. The answer is always the same. Suggest why the data is not given as the total number of people, and that is to account for the different group sizes, or that they are different number of people of different ages, or you can say so the different group sizes can be compared. Figure 10 is repeated below. Again, same graph. Describe two trends shown in figure 10. So pattern, what patterns can you see? Use only the data for the number of people with skin cancer per 100,000 population. So you just need to look at the lines, the dashed line and the bold line. Okay, so you can see that the number of males and female per 100,000 population increases with age. I can simplify that. I can say the number increases with age. Okay, so the number of people with skin cancer increases with age. I don't need to use all of these lines. Keep it nice and simple. What else can we see? We can see that in, so we're looking at the bold line, in females the number increases at a steady rate. We're not seeing any dramatic rises like we are with the males. So in females, the number increases at a steady rate. That should be enough for 2 marks. 2 bullet points, 2 marks. 5.6, remember your biology paper should have or will have 10% maths. So hence here we have a maths question. So the estimated population of males aged 80 to 84 years was 694,000. Calculate the number of males aged 80 to 84 years with skin cancer in that year. Okay, so we're looking at the bold line. Okay, so use figure 10, give your answer 3 significant figures, right? So what we need to do is we need to look at how many males do we have in that category. So 80 to 84, the shaded bar is the males. So I'm going to go to the top of this line because that line is males. And I'm going to read this value off. So it doesn't matter whether, so there's the start of the bar, there's the end of the bar. It doesn't matter which value you read off. Okay, so I'm going to go to the top here and I'm going to read it as 118. Okay, or you can read it as 116 or anything in between. So it's, and be careful, it's per 100,000. Okay, so I'm going to take 118. Total population is that 694,000, and it's because it's per 100,000. I'm going to divide by 100,000. So 118 times 694,000, divide that by 100,000. That gives me 81892. The question says 3 significant figures, so that'll be 819. Okay. So next question, so moving on. So this question is about the heart. Why is the heart described as an organ? So remember, this is your B2 organization topic. So it's described as an organ because it is made up of tissues. The valves in the heart keep the blood flowing through the heart in one direction. Figure 11 shows a heart with one of the valves labelled valve. Explain the effects on a person if the valve labelled in figure 11 develops a leak. A leak is, well, a leak develops when the valve does not close properly. So if that doesn't close, think, what would happen to the flow of blood. So your first bullet point should obviously tell me what would happen. So some blood, but not all, so some blood will flow back into the ventricle. That means, some resident explain question, tell me the science behind it. Yeah, what will happen? Less oxygenated blood would be pumped to the body. So what's the effect of that? Less oxygen being delivered to the cells. We can just say less aerobic respiration. So if there's less aerobic respiration, that means that there's less energy being transferred. You can write that down, or you can just say that the person would become or would be tired. As it does say the effect, the person would be tired, or you can say the person would be out of breath. Final question, faulty heart valves can be replaced using biological or mechanical valves. So biological coming from living organism, living things, mechanical, man-made. The faulty valve is replaced during an operation. Biological valves are from animals or human donors, allow blood to flow through them normally, wear out and stiffen over time, so may need to be replaced. Mechanical valves are made from synthetic materials, so man-made materials. You know, you can have like a type of plastic, type of rubber. May cause blood clots on the surface of the valve, require anti-clotting drugs for the rest of the patient's life, can last for a very long time. A young woman enjoys extreme sports and would like to start a family. The woman needs a heart valve replacing. Describe the advantages and disadvantages for this young woman of having a biological heart valve instead of a mechanical. Okay, so we're writing about the advantages of having biological heart valve. So advantages, and again, everything is there. So use that information. There's a reason why they have given you that information. So advantages, reduces blood clots, reduced risk of blood clots. Okay, because here it says, oh, may cause blood clots. So reduce risk of blood clots with the biological valve. And remember, it does say here that she wants to start a family, so you can say, or you can say reduce risk of blood clots during pregnancy. Doesn't need to take anti-clotting drugs. Because it's a biological valve, you can also say there's a lower risk of rejection. You only need 2 bullet points for advantages, 2 for disadvantages. So if I just quickly take you through anything else that you were thinking of, which potentially could be a mark. So you can say lower risk of rejection. She doesn't need to carry drugs when she's traveling. There's no risk to fetus, because obviously she's not taking any anti-clotting drugs. Disadvantages. May be rejected. Because it is either from animals or humans, so may be rejected, may have to go through surgery more than once, because as I say, wears out, stiffens over time, so may need to be replaced. So for my second bullet point, I would say may have to go through surgery twice, or may have to have additional surgery. it's a biological valve, and if it's coming from a human donor, she might need to wait for a donor. She will have to take immunosuppressant drugs for the rest of her life. And what you can also mention is if the biological valve is coming from an animal, you can say that an animal might be killed. So that can also get you a mark. Okay, so that brings us to the end of this paper. Okay, I hope you found that useful. On through the grade boundaries. So like I said earlier, this was a 2022 paper. The grade boundaries more or less staying in line with the previous year. So if I just quickly take you through the 2024 grade boundaries. So for the biology, so if you were doing this paper while I was doing it, 50 and above. So if you marked your paper, you got 50 and above out of 70, that's a grade 9. 44 to 49 is a 8. 39 to 43 is a 7. 31 to 38 is a 6. 23 to 30 is a 5. And a 15 to 22 is a 4. Okay, the lowest grade you can get on a higher tier paper is a grade 4-3. If it's below that, it will be ungraded. Okay, so what I suggest is the safest thing to do is, it's out of 70, try to hit at least half marks, which is 35. If you get 35, that would be a grade 6. So always bear that in mind. Half marks, more or less, will get you a grade 6. And if you do that for all 6 papers, you should be able to get a grade 6 overall. So, speak to you soon. Take care, everyone.